Hello, I'm
Hi.
Dr. Angie Michaiel, and I'm a science program fellow with the Kavli Foundation. And today we are speaking with Dr. Rosalyn Putland, who is going to tell us about some of her research that she did with the Grass Foundation at the Marine Biological Laboratory in Woods Hole. So, Rosalyn, how are you doing?
I'm great. Thank you. Thank you for inviting me to chat today.
My pleasure. So, how about you start by telling us a little bit about your scientific background?
Yeah, so hi everybody. My name is Dr. Rose Putland. I am an underwater acoustician at the Centre for Environment and Fisheries in the UK. And this summer I was a grass fellow at the Marine Biological Lab. And yeah, had a great summer out there working on the hearing of cuttlefish and squid.
Cool. So, could you tell us a little bit about the Grass Fellowship and then kind of what that meant for you? And then we can go into the science.
So the Grass Fellowship is a 14-week program. It's for independent researchers to come and do a scientific question that maybe they couldn't do at their home institution. So for me, on a day-to-day basis, most of my work is on a computer. I'm very much desk-based now in my role, but I do a lot of passive acoustic monitoring of sounds in and around our oceans. So whether that be geological sound, biological sound, or human-produced sound as well. And this summer I was trying to answer a research question that was on my kind of one of my passions or hidden passions, which is on cephalopods, because they're kind of this unexplored animal when it comes to noise. We kind of speculated for over 100 years on whether they can hear and how they hear that, what that mechanism is. And so I was looking at these two different species, the bobtail squid, which I think we have a cute little video of or a photograph of to show you. Yeah, so this is one of my juveniles. So as you can see from this image, it's less than 2 centimeters in length. Really cute animals to work with. And this is just one of the groups I had. I worked on juveniles all the way through to adults and the pre-senescent adults as well. And this video is just showing when I was doing some behavioral observations of these guys. So putting it into my experimental tank and monitoring their ventilation rates over time. This is either just in control or in response to sound. So my big question, I guess, was, can these animals hear? And do they respond to human-produced sound that's found in their ocean environments?
So, what kind of methods did you use to study that question?
So what's being shown from this video is doing kind of behavioral observations. So with the help of actually one of the other fellows, I was able to set up this video system under IR light to monitor their behavior. But the large majority of my work, which is shown in this picture here, is actually I was doing electrophysiological recordings. So I used it was kind of like an ERG. If you go to the doctors and you have your hearing tested, we can press a button and show our response to a particular sound cue. Whereas in this image, I'm showing kind of my experimental design where we were kind of recording their ether's response to sound cues. So just at the bottom of the image, you can see an underwater speaker that's producing a variety of different frequencies to these animals.
So, when you were doing these experiments, what were you expecting that you would find?
That is a really good question, because I think when you come into anything like this, working with animals, expect the unexpected. You don't know if they're going to actually be able to hear what you're speculating. So I was playing them sounds between 100 hertz and 1,000 hertz. But human hearing's anywhere between 10 and 10,000 normally, or 20 to 20,000 if you're really good at hearing. And so I was playing them a really small range, different tones, to see what they would respond to. And I was actually pretty lucky that the hearing was dead on. They were able to hear within that frequency range with their peak hearing sensitivity around 300 hertz. So this is really low frequency sound for anyone who has listened to animals before, and it directly actually overlaps the sounds that are produced in the oceans from vessel activity, from construction, from harbour developments. And so that's kind of from my day-to-day life when I'm in my management role, thinking about that bigger picture of the effect of noise in our oceans.
So could you tell me a little bit about the results that you found from the electrophysiological experiments?
Yeah, so we have a couple slides that I've put together. So if you can pull those up, that'd be great. So this is kind of just an overview of my original objectives. So kind of are cephalopods impacted by anthropogenic noise and the different ages that I was testing. So I mentioned earlier the juveniles all the way through to the late stage adults, as well as a different species, the stumpy cuttlefish, which I just tested in the adult range. So the next slide actually shows some of my results. So you take these ethers recordings over a different range of intensities of sound as well as frequencies. And all together, I put it on a very simplified diagram here. The two species that I tested, Euphragmidaria, or the bobtail squid, and Sepia bandensis, or the stumpy cuttlefish, in blue. And their frequencies of hearing is anywhere between 100 and 1,000 hertz. Whereas when you compare this to other species, such as the longfin squid or Dorotutheus pelei, it's very similar, right? Kind of overlaps directly with that range. And then the bottom two species are the common cuttlefish, Sepia lesionica, and the common octopus, Octopus vulgaris. And on here, you can see that they've got a little bit higher frequency range and much more limited than the ones I was testing. But again, I was kind of showing that there's that overlap with human activity, as well as the biological and geological sounds we find in our ocean. So they potentially are using this hearing in their day-to-day lives, but it could be impacted. Kind of one of the key results that I haven't shown a graph from here because it's currently unpublished, so I don't want to put that up today, is the hearing that we tested. We did it following noise exposure, and it showed a reduction in their hearing ability. So there was actually a temporary threshold shift in their hearing. So we were potentially deafening them for a short period of time. Of course, that sounds a little bit negative, but the good news story from my data this summer was I retested some of the animals that I was working with a couple hours later. And I actually found that their hearing came back. And that is one of these things that I found most extraordinary from my summer, is that cephalopods could have this ability to regenerate or repair their hearing ability very quickly. And this is fantastic from a neurophysiological point of view, because we want to know more about how this is happening and hair cell repair.
So do you think that the damage that happens to their hearing is that due to just the fact that it's like loud noises, or do you think it's an active process of their hearing being shut down somehow and then being recovered later?
Fantastic question. And I think for me, it was more that it could be this complete shutdown of their hearing. It could be that over time they've developed this evolutionary process that they are protecting themselves from a loud sound source. Over time, way back when in history, animals would be exposed to loud sources from earthquakes, from huge storm events. And so if they're exposed to these loud sounds, they then have to repair their hearing, right? Fishes have also been found to do this if they can regenerate their hair cells in different ways along their lateral line system, as well as potentially in their inner ear. And so this could be a new group of animals that we may have found could also undergo some sort of hair cell regeneration or repair following noise exposure.
So fascinating.
Yeah.
Well, I see that we got a question before about how could you hum or sing the approximate sound that you were playing to the
Well, I don't know if I'm a great choral voice expert, but it's a low frequency tone, like a low hum, so sound, and then kind of a higher sound would be a sound. So that's kind of my fun way of explaining it. Yeah.
Awesome. Okay, so could you tell us a little bit about why cephalopods are interesting to you? Why would you do this work in cephalopods versus any other type of animal or traditional model organism?
Yeah, so in terms of in the aquatic environment, we know quite a lot about marine mammals and fish species. Less is known about invertebrates, and the cephalopods are this amazing, intriguing group of animals. I mean, I know I'm a little bit biased, but they have 3 hearts, 2 gills, blue blood. Their esophagus runs through the center of their brain. They can regenerate limbs. These are just some of the fun facts. And of course, what is most known for them is their camouflage abilities. they can blend into their environment in less than 0.3 of a second. And the Marine Biological Lab, where I was at for the grass fellowship this summer, has some fantastic researchers who are based there year round. And it's really been this pioneering institute for cephalopod work kind of over the years. I mean, back there, they discovered some of the action potentials from giant squid, from the giant axon in squid. And so, that won the Nobel Prize for Hodgson and Huxley a couple of years later. So it was great to kind of be part of this established network of people working on cephalopod research. And I should mention that, myself, I was one of the grass fellows working on cephalopods, but there was 3 others that were also looking at different aspects of cephalopod research, whether it be whether they actually sleep. They were doing some behavioral studies on that. They were also looking at kind of the different parts of the brain, doing various imaging techniques, as well as these behavioral observations on the cultured animals that they have at that facility.
So I noticed you said something about imaging techniques. I'm wondering if you did any imaging in during your fellowship.
I did. At the Marine Biological Lab, it's a fantastic facility for imaging. So this video here is actually showing some light sheet microscopy, and it's showing various sections. So we're able to get a 3D perspective of the inner ear structure. So within cephalopods, it's called a statocyst. It has a central calcareous stone called a statolith, and then various hair cell membranes around it called the cupula Christi. And what this video is showing is DAPI in blue and acylated tubulin in yellow that we could actually use as a marker for the hair cells within this structure. So this is some kind of new techniques and ongoing work to analyze this data. This is kind of rough and ready, putting the images together for sort of to show you today. But yeah, going to keep looking at these images and begin to identify different areas of hair cell bundles in these structures.
So I'm wondering if you did these experiments with animals that were of different ages.
Yes, so I did the imaging on different ages, the hearing tests on different ages, and what was generally found in the results was that there wasn't a huge change in the hearing capability. As is often found in humans, we lose our hearing over time, particularly in the high and the low frequencies. But I was generally getting that they were still able to hear throughout their life, and that they were hearing right from the early ages. So some of the juveniles I was working with were sort of 30, 35 days old, and they've got this great hearing capability already.
Wow, that's fascinating. So it sounds like the cephalopod brain or nervous system in general is kind of this alien system. So it seems really exciting to explore any of those capabilities that we just have no idea what they are right now.
Yeah, as I say, cephalopods are just a great animal to work with. Equally slightly frustrating because we don't know a lot about them. Like there's still so many mysteries that have yet to be resolved. And I hope that many scientists will continue using these as a model organism to find out some of those questions.
So I have one last kind of fun question. So if you had a magic wand and you could do any experiment or solve any scientific mystery, what would that be?
Such a fun question, and I'm definitely going to bring that up the next time I see some of my fellow grass fellows from the summer to ask them that, because it didn't come up in conversation over the summer. For me, one thing we don't know, and this is going to be a cephalopod related one, unfortunately, is we don't know much about the giant squid that lives in the bottom of the ocean. So I would love to send some cameras down there and find out what the heck is going on. Are they as amazing and as big and as dangerous as everybody suspects they are? Like, are they the Kraken of the sea?
How big do you think they would be if they exist?
I think the possibilities are endless. They could be the same size as a sperm whale for all we know.
Wow.
Yeah.
That would be terrifying.
Truly.
Well, thank you, Rosalyn, for sharing your work with us and sharing your experience at MBL during your grass fellowship. Really happy we got the chance to talk. Thanks for your time.
Thank you.